Greetings of the day to everyone. Let me first introduce myself. I am Dr. Shobhna Kapoor. I'm an assistant professor at the Department of Chemistry at IIT Bombay. And I first would like to thank all the organizers of this wonderful symposium to giving us a platform to talk about my research work. With this, let me first take you through our research activities. So we work in the field of lipid chemical biology and membrane biophysics. And there are three main research themes of our group as depicted on the slide. Today, I will be talking about the first subtheme, which is mechanisms of pathogenic lipids in diseases. So with this background, to get everyone on board, let me first tell you that a pathogen is any agent that is capable of causing an infection or a disease. And anything that is derived from these organisms are known as pathogenic in nature, which is also capable of eliciting some kind of infection. In our case, we work with pathogenic lipids from Mycobacterium tuberculosis, which is the agent that causes TB. And what I show you on the right-hand side is a unique cell envelope or lipid membrane structure of this organism, which is very complex. And of most important or importance to us are the lipids which are present in this outermost layer as free, non-covalently attached lipids. There is a notion in the field that these lipids can be secreted by the microorganism during the course of infection. And we basically were interested in exploring the molecular details of interaction of these lipids with the host cell, particularly the host cell membrane, which is the first point of contact with the host cell, which is basically the target cell where the pathogen is capable of causing some kind of an infection. So first, what we did was we designed some kind of a lab infection model wherein we extract the lipids of these mycobacterial origin from the outermost layer, and I show you a representative structure of one of the lipid known as sulfolipid. It is a tetraacylated lipid molecule that has 4 long hydrocarbon chains and a sulfated trehalose head group. So what we did was we basically designed some kind of an assay wherein when we expose these lipids to the host cell, we basically wanted to study its interaction with this cell, particularly whether these lipids are capable of inserting in the membrane, and if they are able to insert, what changes does this interaction bring about in membrane organization and dynamics. So first, we were able to confirm that these exogenously added lipids are indeed incorporated in the target cell membranes by use of TLC as well as mass, wherein we were able to detect signature signals of these lipids compared to the purified control in the treated host cell membranes. So with this in mind, we then asked the next question, whether this insertion can bring about some change in plasma membrane organization. For that, we used a technique known as Laurent two-photon microscopy. So what this technique basically measures is that it uses a Laurent dye, as shown here on the left-hand side, which basically exhibits solvochromic shifts depending on its environment polarity. So this dye, because of its amphiphilic nature, can insert in the membrane. However, depending upon the environment polarity of its surrounding, it exhibits either a emission maxima at a lower wavelength, wherein the membrane is tightly packed and low polarity, or when the membrane environment is more loosely packed, fluid, or disordered, there is more water molecules around the dye, and therefore the emission maxima of the dye shifts by about 50 nanometer. So using the intensity ratiometric mapping of this intensity, we generate a parameter known as generalized polarization, or GP. And this GP theoretically can take values from -1 to +1. Minus negative values basically mean that the Laurent dye is experiencing a scenario which is depicted here, that is, the membrane is more disordered and fluid or high polarity, whereas positive GP values signify a scenario which is wherein the dye is captured in a membrane which is very tightly packed and hence has very low polarity or less number of water molecules around it. So what we did was we took our live THP1 macrophages, which is one of the most extensively used model cell line for tuberculosis research, and we incubated this live cells with this dye. And what you see here is a pseudo-colored GP map, wherein each and every pixel of this image depicts a particular GP value. And what instantaneously you can see is that the cell surface or the plasma membrane of these live cell is not uniform, but rather it has some kind of these orange colored regions as shown here with the white arrow, which are basically high GP areas, or in other words, these are lipid domains of very low polarity or very compact in nature. In addition to mapping visually, visualizing the cell surface organization, we were also able to extract these pixels and plot them in the form of histograms, where you can see at least two distinct populations of lipid domains, wherein one has a high GP value and one has a low GP value. So with this method, we were able to see that the cell surface or plasma membrane of live cells depict a lipid domain organization or phase segregation of lipids on the cell surface. And why this is important is because, in accordance with literature, the plasma membrane of mammalian cells is not a uniform bilayer. Rather, it exhibits some kind of domains or nanoclustering of lipids, which has some kind of a functional implication. And it is one of the defining feature of plasma membrane in mammalian cells. What you see here is a 3D projection of a high super resolution image of a mammalian cell, wherein you can see these small and big hills, which are basically nothing but lipid domains of variable sizes and polarity. And these domains, which are yellowish to red, are basically very tight, compact lipid domains, which have very low polarity and very ordered in nature. And why having such kind of a domains is important for the cell is because these regions actually act as platforms to recruit or sequester membrane proteins, which basically act as a channel between the outside and the inside world for communication in the form of cellular signaling, which in turn regulates a lot of cellular processes in cells. And the logic we had was that any agent that can disorganize such kind of a lipid domain organization might have an implication on cellular functions. So with this background, we basically decided to explore whether mycobacterial lipids, if they are capable of causing some kind of remodeling of the plasma membrane domain organization. And what we found using our optimized two-photon microscopy, that indeed it does. Compared to the control cell seen on the left-hand side, what we found was after interaction with the sulfoglycolipid 1, the membranes of these live cells showed a complete reorganization of its lipid domains, wherein no more these orange colored regions were visible, but rather the GP counts of the entire image was shifted to lower values, suggesting that there is a fluidization effect caused by the insertion of this mycobacterial lipid. In addition, we were also able to quantify that indeed one population, as seen in the pictorial representation, also decreases in a concentration dependent manner. So with this, we were able to actually conclude that the host cell lateral membrane organization is remodeled by mycobacterial sulfoglycolipid. Then what we did next was that the images that I showed you, they basically had information about all the membranes, not only plasma membrane, but also internal membranes, because this dye is capable of internalization. So using the same method, what we did was we did some kind of an image segmentation, wherein we now only look at the plasma membrane of these cells with and without this exogenous lipid molecule. And what we were able to see is that in addition to the global fluidization effect, the plasma membrane, although less, but also shows a similar remodeling by this mycobacterial glycolipid. Then next, what we asked was that whether this kind of modulation of the membrane organization is selective to sulfoglycolipid 1, or whether mycobacterial lipids of other let's say structures are also capable of affecting the plasma membrane organization. So we applied the same method, and we were quite happy to see that indeed this remodeling of the plasma membrane is very selective to the nature of the lipids that we chose, wherein compared to sulfoglycolipid 1, other lipids such as PDIM and LAM were not able to show a similar fluidization or disordering of the plasma membrane. And that underscores that these Mtb virulent lipids, when they interact with the host cell membrane, they have a selective effect on the remodeling of the plasma membrane. The next question then we asked was, okay, there is a reorganization of the plasma membrane, but what about the membrane dynamics? For example, does this organization is concomitant with a modulation of the diffusion of plasma membrane components? So for that, we used fluorescence recovery after photobleaching, and we used two different probes. The first probe, as shown on the left-hand side, is a probe that has all saturated chains so that it partitions only into the ordered regions of the plasma membrane, whereas the probe, which is shown on the lower right-hand side, has unsaturated chain, and therefore it has higher affinity towards more fluid, disordered regions of the membrane. So with these probes in hand, we basically tested the diffusion, or calculated the diffusion of these dyes in the control as well as in cells which were treated with different structurally diverse mycobacterial lipids. And what we were able to show was that whereas sulfur glycolipid 1 increases the diffusion in the two different domains, but other lipids, for example TDIM and PDIM, they try to reduce the diffusion of the plasma membrane components, again suggesting that these mycobacterial lipids have a different effect on the lateral lipid organization as well as mobility of the plasma membrane components. Then what we decided going further was whether these changes in the organization and dynamics are also reflected in the membrane mechanics. So for that, we used atomic force spectroscopy. And as shown in the cartoon representation, what we do here is that we indent the surface of these live cells with the help of an AFM cantilever. And during this indentation, you basically record force versus distance, as shown in this black curve. And by analysis of this black curve, we are able to calculate parameters of elastic modulus or stiffness. And what we found with sulfur glycolipid 1, that the stiffness of this live cell membrane was decreased, suggesting that there is some kind of a softening of the membrane. And if you put it in context of infection, it becomes very clear that, okay, when the bacteria is in kind of close contact with its target cell, it secretes these lipids. These lipid inserts in the plasma membrane, they soften the membrane so that the pathogen uptake can be facilitated. In addition to that, we also measured tether spectroscopy, wherein if you look at this force versus distance curve, when you indent the surface of the cell with the help of an AFM cantilever, you measure the black curve, which is basically your gives you an estimate of your elasticity. But when you retract this AFM TIM, then thin membrane nanotubes form between the tip of the cantilever and the plasma membrane. And these basically appear as regions of constant force in a force distance or extension curve. And pictorially, they would be depicted as shown here, wherein we have, let's say, different numbers of membrane tethers as the membrane is being pulled by this AFM tip. And the parameters that we obtain from here is the tether force associated with the membrane stiffness, tether length, as well as the number of the tethers. So what we found with sulfur glycolipid 1 was that the tether force was decreased, which means that the SL1 treated cell membranes require less force to be extended, and their number were found to increase. And in general, the formation of membrane tether is associated with formation of or induction of curved regions. So there is a tantalizing possibility that SL1 insertion into the membrane can lead to the formation of such curved regions, which affects the membrane nanotube formation as well as their dynamics. Next, we kind of extended these studies with different structurally unrelated lipids. And what we found was that different lipids were able to modulate the elastic modulus as well as the tether dynamics to a different extent, which eventually can be mapped to their structural aspects. For example, PDIM having saturated chains is likely to pack very tightly, and that causes increased stiffness, whereas a lot of cyclopropane rings as well as the methyl branches in SL1 can mitigate this stiffness or this tight packing and lead to reduction in the stiffness of the membrane. So the take-home message, what we obtained was that all structurally unrelated mycobacterial lipids affect membrane elastic modulus and tether dynamics differently. The last thing what we wanted to explore was whether this membrane remodeling in terms of organization and dynamics that we see, is it also percolating to the actin cytoskeleton? So actin cytoskeleton is nothing but a network of actin protein which is just beneath the plasma membrane, and it provides a much needed force and shape to the cell body, and it controls a lot of different cellular processes such as migration and differentiation. And it has been found that pathogens, including mycobacteria, can actually modulate this actin network to facilitate entry. So we wanted to basically see whether the mycobacterial lipids are one of the factors that can affect or bring about change in this organization. So we used confocal microscopy and used antibodies that are capable of staining these actin filaments inside. And in absence of these lipids and in presence of different kinds of mycobacterial lipids, we quantified first the abundance of actin, which we found was modulated to different extents, wherein some of the lipids were able to cause an increase in the actin abundance, the others did not cause much change. In addition to the abundance, we were also able to see that the actin morphology is also altered to different extents. For example, in normal conditions, these cell types exhibit three kinds of actin morphologies, either filaments or puncta, which is, these kind of small dots, or patches, which you can see here. And what we found was that when the cells are incubated with these mycobacterial glycolipids, these different actin morphologies redistribute in different amounts. So clearly, there is a role of mycobacterial lipids in remodeling not only the membrane, but also the associated actin cytoskeleton. And although bacteria-induced actin fragmentation has been shown, but our work reveals that the MTB lipids may act as one of the key mediators of this process. So what basically we concluded altogether is that these structurally diverse, unrelated mycobacterial glycolipids are capable of inserting in the plasma membrane of a host cell, wherein it brings about changes in its organization and mechanics. It affects the diffusion of plasma membrane components, which we have only explored proteins up till now. It also leads to actin disruption. And what I was not able to show you is that the functional significance of these interactions actually culminates by seeing increased cytokine and autophagy signaling, which basically brings us to the mechanism that we are proposing, wherein this organism can fine-tune its interaction with the host cells by modulating abundance of these lipids, because each of these lipids produces a distinct effect in different aspects. And this unique conformational and structural makeup of these lipids regulate host membrane structure and associated signaling functions as well. So with this, I would like to conclude my talk. I would like to acknowledge my group members for their hard work, my institute for providing me all the facilities and a good collegial environment, my collaborators, all my funding sources, and finally you for your kind attention. Thank you. I'll be happy to take any questions.